No explanation for orphan genes. But 20 to 30% of protein coding genes of animals in species have no similarity to other animals. This defies evolution. It's not small incremental change. It's a big problem for Darwinism. And some examples of this are antifreeze in fish, genes in humans for neural development. 12% of the fruit fly are de novo genes, or orphan genes. The author of the study went on to acknowledge that invoking this mechanism poses a severe problem for the evolutionary theory, since it doesn't really explain the origin of any of its non-trivial requirements for functionality. It was another quote. They are the molecular equivalent of a mysterious beast in the depths of a rainforest, a biological enigma seemingly unrelated to anything on Earth. That's what the author of that said. Okay, one more slide. The basic cell. So scientists say, you know, we can't figure out where life comes from, how this all organized. Let's try to work backwards. Let's find the simplest cell that we can. Mycoplasma mycoides. And that's the simplest one that they know of. And it has 900 genes, single cell. They thought, let's try to take a gene out, and we'll see what happens. Let's try to take another one out to see what is the minimum amount of information needed for a cell. Good idea. This was just released like 2 weeks ago. Did anybody hear this on the news? No? You guys aren't listening to science nerd talk? Okay, this was just released a couple of weeks ago. What is the minimum amount of information needed for a living cell? They got it from 900 to 473. That's as far as they could go. 30% of the 473, the function is still unknown. All they know is if they take it out, the cell doesn't work. Okay, so one more slide. So these are the things that they decided you needed to have, the genes in there, to have any kind of functionality in a cell. You have to be able to copy your DNA, because if you can't have offspring. You have to be able to transcribe and translate it into protein. You have to divide the cell. You have to make everything that you cannot get from the environment. You have to have cell maintenance. Is that irreducibly complex? I think that's probably the definition of it. I think that's very irreducibly complex. One more slide. Scientific American said in 2001, don't tell the creationists, but scientists don't have a clue how life began. How fundamental innovations arise in evolution is one of the most enigmatic, fundamental mysteries of nature. I was just watching a video the other day. I showed it to Mickey, and this professor from Rice University, Rice? Yeah. Is that what it was? Rice University. This guy makes nanomachines out of molecules. Him and a team of scientists made cars out of molecules, microscopic cars. And he was explaining how they did this and how difficult it was. And the process, in order to make one little part of this car, you had to somehow get the molecules to come together. And then in order to do that, you had to add this chemical. Then you had to stir it quickly. And then you had to get out the bad chemicals. And then you had to heat it up to 300 degrees. Then you had to cool it down to minus 40 below zero. Then you had to filter it. Then you had to add another reagent. And he's going on and on about this. And he's saying, there is no way that this could happen in a mud puddle in a cave. That's what he said. There's no way. He says, Mickey can vouch for this, because he just watched this today. He said, any scientist that tells you that any scientist has an understanding of how life began doesn't know what they're talking about. Okay? And then he goes, he's very colorful, then he goes, they don't know what they're talking about. Okay? Because he was explaining how difficult it is to get these chemicals together. Okay. Next slide. This is new too. Some of this is new. Sponges are studied too, because sponges, they think, were the first animal. And an animal being defined as a group of cells that works together. Okay, and so this study, this new finding showed that sponges are 640 million years old. They found chemicals in rock of that age that only sponges make. Okay, so that's how they figured that out. So that they were on the earth then, at least that's what the thinking is. And so it has 18,000 genes, and it's supposed to be the earliest common ancestor for animals. There are pre-adapted, this is the freak out part, there are pre-adapted genes for muscles and neurons. Okay, so in the earliest common ancestor, there are genes in sponges for muscles and neurons. How and why did those get there? They call them pre-adapted. Now, pre-adapted sounds way too teleological for biologists, and so they changed the name to X-adaptive, or co-option genes, because pre-adapted just doesn't sound like, that's saying, hey, wait a minute. So genes that stop cancer are in sponges already. At that new findings, I spread that, they are the first animals to inhabit the earth. And then this is another new one that just came out, because they study these like crazy. They've been trying for years to figure out how sponges make their skeleton. And they make them by these little spicules, which are like little spears, I guess, is the best way to describe it. They found out that the spicules are manufactured by certain cells in the sponge. Then they are transported to where they need to go by different cells. Then a new group of cells pierces the area where they need to go, and then a new group of cells raise it into position and cement it in place. That's the earliest living animal that they know of. That's how they build their skeleton. Okay, now how can that possibly happen by gradual, slow change? It can't. Next slide. I'm almost finished. I promise. The Cambrian Explosion. Most people don't know about this, and I was surprised when I read about it, but there are groups of animals in biology, large groups of animals. 20 of the 23 groups of phyla in the animal kingdom appeared in the Cambrian era, in a span of within about 10 million years, okay, which is a blink of the eye in geologic time. We're talking 550 million years ago. All of a sudden, in the fossil record, 20 of the 23 animals appeared. Okay, before that, there were no animals with 2 arms and 2 legs. No bilateral animals before then. They thought, well, the fossil record, we just can't see it yet, all of it. We'll get it eventually. We're going to find those fossils that were there before. And they found good areas where fossils should develop, but they're not there. Darwin knew about this problem with the Cambrian, and he spoke about it, saying that that was a major issue, that if it couldn't be resolved, that was something that could throw his theory into chaos. So that's called the Cambrian Explosion. Next slide. If the Cambrian Explosion, in order for that to happen, these are the things that you would need in order to develop those new animals. This enzyme for ligaments and tendons, hemoglobin, glass skeletons, eye formation, Hox genes are the early embryonic genes for body plans. 429 protein families are common to all animals, or common to all eukaryotes. 136 are common to animals. Those would have to have arisen in the Cambrian time. You also need regulatory genes and enzymes for all these proteins. Next slide. So biologists are changing the terminology because of all these problems. Descent with modification is becoming species-specific biology. And in studies, they're changing how the language is because it doesn't reflect what's going on. Random mutation is becoming directed mutation. Pre-adapted genes, that's X-adaptic, I think is what that one is. Junk, as far as junk genes and de novo genes, now when you read the literature, they change and they say, that's exactly what we'd expect from evolution. Because evolution is so powerful that it can make de novo genes out of nothing. Okay, that's what the people that are still sticking with the neo-Darwinism, they're changing the language. Evolutionary tree is now, they have to separate it because there is no evolutionary tree. There's a molecular tree, which does not match up with the morphologic tree or the molecular clock tree. Those don't match up. So when they talk about it, they have to talk about this one or that one or that one. The evolutionary mechanism is becoming the unknown mechanism. They say that all the time. It's the unknown mechanism, when they try to explain what happens here. Next slide. So there is no materialistic cause or evolutionary mechanism that has demonstrated the power to produce large amounts of specified information. Intelligent causes have demonstrated to produce large amounts of specified information. Intelligent design is the best explanation. Next slide. Thank you.
Just one more thing that Dr. Tour said today from Rice University, and it was really great. He says, if the chance of what has happened, the origin of life, if that happening by random mutation evolution, is basically like taking the 26 letters of the alphabet and giving them millions and billions of years to come out with war and peace.
Yeah.
And it will never happen. This guy says that, and he went on like Bob said, that these scientists that make these unreasonably claims, he says they really don't know anything. And they're about the origin of life, and they're just trying to fill in the gaps. Now, once again, I thank Dr. Herman for coming out here tonight on a beautiful day and giving this this lecture, this presentation. It was very convincing. Thank you so much for that. We're going to take a 10-minute break, and then we're going to start talking about CNA. So if you want to come back for that, please do. Let's finish with a round of applause.
Thanks for coming, Rob. So, in Harry Potter, when they start pulling memories out and they're like protein strands.
That's what it is. That's exactly what it is.
You can do that.
Maybe not yet.
Or my career teacher, we did. You can teach a rat to circle a snake and get another rat.
To run a maze.
Touch a little more red light.
Okay.
To do what?
Okay? Touch a marker. Oh, red light comes on. If you do that, if you do it consistently for 21 months, you sacrifice that rat. And you take RNA out of it. Oh, good. Okay. And then you create another rat. Beyond infancy. You can actually just inject that into the brain. Clean that thing, he'll learn from one at the time.
Yeah, that's what they're talking about. That the mechanism is built in. It's not something that changes through the What's that? Yeah. Yeah.
They're saying it's basic. Having a day your friend, his son, his mom.
Yeah.
So whenever you get an argument, because we're talking about some of these polypeptides.
Yeah. That's what. Yeah. It's polypeptide.
We can make polypeptides.
Right. Right.
Ask somebody at the point where you can make polypeptides. Polypeptides are not live more than microseconds outside the cell.
Yeah.
So these polypeptides are in our cell.
Yeah. They're not stable in water.
Where did they
They're not
start
stable in water.
if they're in the cell?
Right.
That's the one we got out of.
Right. Yeah, and there's a lot of disagreement between the professions. Because the biologists, and Dr. Tor was talking about this, the biologists have their theories on this. And then the chemist or the mathematician comes along and says, wait a minute, there's no way that's going to work. And they say, listen to that theory. Then what they do is they go and erase it on the blackboard and they put something else up there that's just as unlike it. And he said, so.
One question, again, the question of if evolution is there and we came from monkeys, why is that monkeys?
Uh-huh. Yeah, you would think it would all evolve, or it would all.
And then we know that the chimpanzee why hasn't came out of that same.
They have been mutating different ways, mutating fruit flies
That's
for
it.
about 60
Bob,
years now.
you're still recording.
Am I? Oh, okay. For about 60 years now. Just push the red one there. And,
Push that red